Hello, all NEET aspirants. You are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, the molecular basis of inheritance. The subtopic it is search for genetic material. If you see the chronological order of the development, the factor to the chromosome and genes. In 1865, Mendel proposed the factors, the basis for inheritance. In 1869, almost the same time, Miescher, who discovered nuclein, an acidic substance. But it took long time to decipher the structure of the DNA and to be proven as a genetic material because of some technical limitations in extracting a long intact polymer. But in case of 1926, quest to determine the mechanism of inheritance reached to molecular level. Contribution of Sutton, Boveri, Morgan, Mendel, all their contribution narrowed their search to chromosome level. So there is a development factor to chromosome level, factor that was invisible. Now the chromosomes, which are the carriers of heredity, but chromosome having DNA, RNA, and a protein. What was the real chemical as a genetic material had not been answered. In 1928, the Griffith carried out his experiment in Streptococcus pneumoniae to find out what was the genetic material. In Streptococcus pneumoniae, in his experiment, a miraculous transformation of a bacteria in a physical form due to absorption of something from the surrounding. So, absorption of genetic material from surrounding bacteria, and the bacteria changes into a new form. What do you call it? Is a transformation due to transfer of something. That's something nothing but genetic material. In Streptococcus, there are the two strains. In one strain, it is having a mucus coat. It is virulent, causing pneumonia. In culture media, it develops shiny colonies. The other, it do not have any mucus coat. It is rough. It is non-virulent. Do not cause any pneumonia. In culture medium, it develop into rough colonies. Next, when a strain that is virulent having mucus coat injected into the mice, mice die. When R strain injected into the mice, it alive. When heat-killed A strain injected into mice, it alive. When heat-killed A strain and live R strain injected into the mice, it died. Because after autopsy of the mice done by Griffith, who recovered some live A strain, that means R strain somehow been transformed. R strain transformed by heat-killed A strain due to transfer of something, that is transforming principle genetic material. The transforming principle genetic material uncleared by Griffith. So something transferred from heat-killed S to R strain, which got transformed, R strain got transformed by heat-killed S strain due to transfer of transforming principle, nothing but genetic material, but transforming principle uncleared by Griffith. And biochemical characterization of transforming principle of Griffith experiment done by Avery, MacLeod, McCarthy. Before Avery, MacLeod, McCarthy, they carried out experiment in 1933 to 44. It was thought to be a protein. And Avery, MacLeod, McCarthy carried out experiment by collecting the biochemicals in heat-killed A strain, the DNA, RNA, and a protein. And they took all the enzymes, RNases, proteases, DNases. And RNases is going to digest RNA. So transformation unaffected. So next remaining were protein and DNA only. They took proteases. It digests the protein into amino acid. Transformation unaffected. They took DNA ases. Transformation affected. So DNA was the chemical transforming principle that is responsible for transformation. Because difference between what is the difference between DNA and DNA is DNA is a polynucleotide. DNA is a polypeptide. DNA is hydrolyzed DNA into deoxynucleotide. So DNA was the chemical, biochemical transforming or strain to a strain, but biologists not convinced, not univocally it is accepted. The univocal acceptance by Hershey and Chase experiment, what they call transduction experiment. In earlier Griffith experiment, it is transformation experiment. Now it is transduction experiment. The transduction experiment carried out by Hershey and Chase in 1952. So in earlier DNA versus protein versus RNA. Now it was narrowed DNA versus protein because bacteriophage are viruses which infect bacteria, and bacterial cell treat viral genetic material as if it was its own and makes viral particles. In their experiment, they took P32S35, a radioisotopes, and steps in their experiment transduction, labeling, infection, blending, centrifugation. Both the viruses are labeled with P32S35. P32 labeled into DNA but not protein because DNA having phosphorus. Protein does not have phosphorus. And S35 labeled into protein but not DNA. DNA does not contain sulfur. S35 labeled into protein at cysteine and methionine because it is having sulfur. Now the labeled viruses are made to infect with bacterial cells. There is a multiplication of viruses inside the bacterial cell. As code proteins still attached to the bacterial cells, separated by a blender in a blender, what do you call it is blending. Separation of code protein from bacterial cells, what do you call blending. Next, the viral particle present in the bacterial cells separated by spinning in centrifuge by applying centrifugal force. And they observed S35 not in cells, S35 in supernatant. So as S35 labeled into the protein, they observed in supernatant but not in cell. P32 they observed in bacterial cells but not in supernatant, as P32 labeled into DNA. So DNA was the genetic material, the DNA was only the chemical that entered into the bacteria that was responsible. So it is a univocal proof that DNA was a genetic material. So protein versus DNA, it was resolved with Hershey and Chase experiment in 1952. I am signing off from this episode. We will see in case of the next episode the properties of genetic material and the rest of the topic like RNA world and semi-conservative replication experimental proof.